Arthur Frank Harvey was born on the 21st of March 1910 in Ebbw Vale, South Wales. In 1929 he graduated from the University College of South Wales and Monmouthshire with a BSc in electrical engineering subjects, receiving the Page Gold Medal for the best students in the engineering department. The university described him as being the most brilliant student the department has had for many years, exceptionally brilliant and likely to go far. Dr. Harvey's early career included an apprenticeship at the South Wales Power Company, working as a technical assistant for Johnson & Phillips, and later getting his PhD at Oxford University. In 1960, Dr. Harvey won the IEEE Microwave Prize for his paper Periodic and Guiding Structures at Microwave Frequencies. He also wrote several papers and 2 books on microwaves and lasers. His work included research and development on millimetre wavelengths and coherent light techniques. Dr. Harvey died on the 26th of March 2006, but not without having left a lasting legacy. In 2010, the IET dedicated substantial funds left by Dr. Harvey to the creation of the Dr. A.F. Harvey Trust Fund. Dr. Harvey's terms were simple. The donation must aid the furtherance of scientific research into the fields of medical, microwave, radar or laser engineering. The IET's trustees agreed the funds would be used to offer an annual research prize covering all fields over a 3-year cycle. It is the IET's largest prize fund, with the winner being awarded an engineering research prize of £350,000. The prize fund's Search and Selection Committee searches for potential candidates from around the world, drawing on wide international networks. The committee draws up a shortlist of candidates from whom additional information is requested for further detailed consideration.
We are working on these tiny, small things which are called dielectric nanoantennas. What we are trying to achieve is some elements which can control light, and like that we can generate, for example, holograms which can move. And like that we can produce holographic displays and also some virtual and augmented reality devices.
So it will open some opportunities that we can test things that might be quite difficult in a traditional application to one of the funding agencies. So we can do a lot of new things and use this as an opportunity then to start many other new projects that will nucleate from this research prize.
I'm excited about my work. I love what I do. Every day I try to excel myself, what I did in the past, and we'll see, but it's baby steps, so I hope that in 1 year I will have something exciting to tell.
Dr. Harvey achieved great work in his lifetime. His legacy continues through the prestigious IET A.F. Harvey Engineering Research Prize.
Hi, I'm Danielle George, President of the Institution of Engineering and Technology. Welcome to this year's Harvey Prize lecture called Spinal Cord Gateways to Reverse Paralysis, delivered by our incredible winner of the 2020 A.F. Harvey Engineering Research Prize, Professor Grigory Courtine. I'm using my term in office to champion difference makers, people who are having a massive impact on the world around us, and to inspire more young people into engineering and technology. Today, the IET turns 150 years old, and our anniversary gives us a fantastic opportunity to celebrate our rich history. You just saw a short film showing the remarkable life of Dr. A.F. Harvey and the history of the A.F. Harvey Engineering Research Prize. Today, we'll hear from our 2020 winner, delivering his inspiring lecture, and then afterwards you'll have the chance to ask him some questions as well. The prize, an impressive sum of £350,000, is awarded annually to phenomenal researchers working in radar and microwave, laser and optoelectronics, or in medical engineering. Our research and selection panel look globally to potential candidates each year, drawing on their expansive international networks to reward the most innovative workers in these fields. Professor Grigory Courtine, a neuroscientist and researcher at the Swiss Federal Institute of Technology and the University Hospital Lausanne, is our 10th winner of the prize. His passion for translational neurosciences has fueled his research in the development of neurotechnologies to improve recovery from neurological disorders. After winning the Chancellor Award during his postdoctoral at the University of California, Los Angeles, he established his own laboratory at the University of Zurich in 2008, before joining the Swiss Federal Institute of Technology, Lausanne, in 2012. He is now a full professor of neuroscience and neurotechnology in the Center for Neuroprosthetics at EPFL and in the Department of Neurosurgery at the University Hospital Lausanne, where he's also the director of DefiTech Centre for Interventional Neurotherapies. He's also the Chief Scientific Officer of GTX Medical, a startup he founded in 2014 to translate the neurotechnologies developed in his laboratories into clinical treatments. So huge congratulations, Professor Courtine. You have a wonderful trophy. Many, many congratulations. I'll be back with Professor Courtine after the lecture. So get thinking of any questions, pop them in the chat, and we'll try and answer as many of them as we can.
I wish I could be with you today to tell you the journey that took David and myself to this very special moment of my career. 10 years ago, David had a spinal cord injury that left him paralyzed from the neck down. But I will let David share this traumatic moment of his life with you directly.
This is the accident. Yep. Yes, many years ago.
David had a spinal cord contusion due to this gymnastic accident. And this contusion damaged the pathway coming from the brain, meaning that now when he sends signals from his brain to activate the cell that control muscles, the signal is weakened, leaving him impossible to activate his muscles. And when I started my career, I first modeled this type of spinal cord injury in animals. So we developed this model of severe contusion, which you see here in red, that's going to interrupt almost all the nerve fibers coming from the brain, yet a subset are spared. Not sufficient, however, in order to enable the animal to regain the ability to use his legs. However, we knew for 100 years of research that the neuronal network that normally control leg muscle are located below the injury. They are completely intact, but disconnected from the brain and therefore non-functional. So our idea at the time was very simple: apply electrical stimulation over the lumbar region of the spinal cord that control normally the leg muscle to reactivate the spinal cord. And the result was very surprising. Immediately, an animal paralyzed would start walking. You turn off the stimulation, locomotion stops, but resume immediately when you turn back the stimulation on. So that was all the work I performed when I was a postdoctoral fellow in the time in Los Angeles at UCLA, where I was also a fellow for the Christopher Reeve Foundation. You know, Christopher Reeves, I've been growing up in the 80s, was really an iconic figure for me. And as a young scientist, my dream was really one day to experience that specific moment that was embodied. It was a little commercial, actually, that was displayed during the Super Bowl that I would like to share with you, because that's really crystallized all my dream as a young researcher.
And in the years since the new millennium, the world has seen such progress. In 2004, the tide was turned against AIDS. 2 years later, great strides against cancer. And tonight we celebrate a remarkable breakthrough in spinal cord injuries, made possible by countless researchers and contributors. And to present this award, we have some very special guests.
In the future, so many amazing things will happen in the world. What amazing things can you make happen?
Christopher Reeves was really an inspiration for me, and he continues being an inspiration today. At this time, I thought it was too early to jump to clinical application. So when I started my own laboratory in Zurich, my first goal was really to understand the science underlying the effect of this stimulation. That was very empirical at the time. And having a background in physics, we started by modeling the effect of the stimulation. So we built this very advanced computational model that allows us to visualize in silico the electrical potential induced by the stimulation. And this is combined with this very advanced, precise anatomical model of the nerve fibers, the neurons. And through this simulation, we understood that actually the electrical stimulation recruits a very specific type of fibers that are called the A-alpha nerve fibers. Basically, they are fibers coming from the muscle receptors that encode the length of the muscle. So what it means is that the stimulation does not penetrate the spinal cord, but flows on the side and will activate the large diameter afferents in the dorsal roots. And for us, this prediction was very important because that would mean that this apparently unspecific stimulation could be turned into a very specific stimulation due to the biophysical properties of the spinal cord. Was it true? We had to verify this prediction. So for this, we developed a transgenic model that allows us to visualize this type of nerve fibers over the entire spinal cord. And you can see here in this zoom, these nerve bundles that are going to be depolarized when we apply the stimulation. And in this transgenic animal, we have been able to record the activity of the neurons in what is called the dorsal root ganglion. So that's where we have the sensory neurons. And you can see here the response of all the neurons in the dorsal root ganglion when we stimulate. I can circle the one that are activated. And because it's a transgenic animal, we can superimpose the signal that tell us which one are from this large diameter afferent neurons. And you see that there's almost 100% of them that are what is called proprioceptive neurons. So it looked like it was real. So we went one step further. We created an animal in which we ablated genetically just these large diameter afferent fibers. And as you can see on this video, the animal worked very well with the stimulation when he has an intact system, but without only this small population of nerve fibers, there's absolutely no effect of the electrical stimulation. Really demonstrated that this is the neuronal pathway that we activate with the stimulation. Why is it important? Well, now we can operate a transition from a continuous stimulation of the spinal cord to a very precise, what I call biomimetic stimulation, meaning activate all the dorsal roots with a spatio-temporal timing that will replicate the way the brain naturally activates the spinal cord in order to walk. Well, it was an additional challenge that to do this, you need to have many electrodes very precisely located over the spinal cord. And I was lucky at the time to be collaborating with Stéphanie Lacour, who was also nominated for this prize, and we really teamed up to develop this stretchable electrode array that you can twist, you can stretch, and it still conduct electricity, meaning they can resist for a very long time in a very complex environment of the spinal cord of rats. This technology, breakthrough, allows us to perform this Princept experiment. The rats has this very severe contusion of the spinal cord, it's completely paralyzed. We apply the continuous stimulation, so it's just a tonic activation, and the animal walk. But it's so early after the injury, you will see at some point it will collapse. And this is when we turn on what we call the targeted or biomimetic stimulation that will activate the spinal cord as the brain would do naturally in order to walk. And you can see how we rescue walking and increase the vigor of the leg movement. So that was very important. Now we have an understood evidence-based stimulation paradigm. But I was still frustrated because this stepping is automated in response to the movement of the treadmill belt. That was not voluntary walking. So we had to move away from this paradigm of automated locomotion on the treadmill to have a more natural environment that would encourage the animal to participate in the act of walking. So we've developed this massive robotic structure, 120 kilo, but with soft actuators that allow to decouple the robots from the rats. So the rat does not feel the robot, just supports the animal against the direction of gravity, like a father would hold his young child during the first insecure step. And then we could test, can the animal now initiate walking? At the beginning, it was very frustrating. The animal would walk fantastically on the treadmill, but was incapable of initiating and sustaining walking over ground. But with training, performance improved. And after several months of training, a rat completely paralyzed now can stand, and when they want, initiate this full weight bearing locomotion to sprint toward a delicious piece of Swiss chocolate that we position at the extremity of the runway to motivate the rats. So that was incredible. That was really the breakthrough we wanted to obtain in order to operate the transition to human. And this is indeed this moment when the rat was walking again that I met David. So it was 1 year after his spinal cord injury. He came to the laboratory, he had seen the rats, and he was ready. So let's go ahead and start a clinical trial with me. We published this data. They were broadcasted throughout the planet. I mean, the little rats ended up in the front page of the New York Times, which suddenly triggered a tremendous amount of pressure, you can imagine, on our shoulder to achieve the translation, because we realized that, from the sophisticated environment of the laboratory in rats to a therapy that can help David, there was a tremendous gap that we had to fill. And at the time, I understood that it was very important to find an environment that would be adequate to achieve this translation. This is why I moved the laboratory to the EPFL. This is the Swiss Federal Institute of Technology, so where all the advanced technology are developed. They're amongst one of the best engineering schools. And it is very much linked to the University Hospital of Lausanne. And Newsweek ranked this hospital, the CHUV number 9. And more important for me, there was an amazing neurosurgeon who is used to put electrode in the brain of people with Parkinson's disease, in the spinal cord to treat pain, etc., and who is really willing to commit a lot of her time, energy, and skills in developing new treatment for people. This person is Jocelyn Bloch, and she became an incredible partner ever since. And together we could really prepare the translation in human. First thing, Jocelyn thought, well, we need to be very precise in the way we are going to develop this therapy in humans. So we need to develop adequate technology for the human spinal cord. Yep, we started all over again. This electrode we developed for rats, we needed them for humans. So we use the same pipeline, develop this highly realistic biophysical model of the human spinal cord, build atlas with many, many patients, and then we are able to really, from MRI scan, to have computational model of the spinal cord, of the roots that we know we activate. So what we can do now is, for example, you want to target one given root, then we place a grid over it, we run simulation that tell us where the field is created, where we should put the electrode in order to target this one specific route of the human spinal cord. And then you perform the same for all the different routes that we want to target, because this is our target. We had an electrode array design, and then we use medical grade technologies in order to build this electrode array. Next step is to insert this electrode array over the correct region, what we call the motor hotspot. They're in the middle of your back, in the lumbar region. So for this, Jocelyn needed a very good preoperative planning to know really where she had to position the electrode array. So we used the same computational pipeline to build for each participant these personalized computational models from MRI scans of new generation. And in silico, we can predict where the electrode array should be to target individual posterior routes of the spinal cord to modulate specific region. So it was very important. When we had this, we were ready. It took actually several years in order to transit from David being ready for the implantation to actually this very important day when he became the first participant of our clinical trial. So everybody was here. There was a lot of tension, as you can imagine, on this day. Jocelyn was ready, we were all ready with the team to do all the electrophysiology, make sure the electrode array was at the right location, and it went super well. Then we had to tune the stimulation. So that's how it works. Simply, let's focus on just one movement. For example, here, asking to do a full leg extension, so activate the quadriceps muscle. But here it's completely paralyzed, so participant is completely incapable of activating this muscle. We have functional MRI that tell us which region of the spinal cord is actually responsible for activating this quadriceps muscle. So we are going to target this region. Remember, we activate the dorsal roots. So this is the L3 dorsal root, the third lumbar posterior root. So now we have our electrode array. We create a little configuration that will steer the electrical potential toward these roots. And when we do this, immediately, but when the patient desires, he activates the quadriceps muscle and performs this full leg extension against the direction of gravity. That's one joint. Now imagine if you want to walk, of course you have to reproduce, this very complex symphony of activation of many different muscles. So you need to orchestrate this stimulation of many posterior roots with a very precise timing. That's really what I like to call the precision of a switch watchmaker, because this is so precise, with a 20 to 40 millisecond precision in order to achieve this. And I'm going to tell you really how it works so you really understand. That's David. If you want to walk, normally you activate your muscle with a very specific timing. You can see here the normal sequence of activation of the muscle. And as I told you, we know where are the neurons that control these different muscles because we have functional MRI in each participant. So if we want, for example, to facilitate the first phase, this flexion of the leg, then we know that these muscles are located in the upper part and the lower part of the spinal cord, hip flexor and ankle flexor. So up with our electrode, we're going to target these two posterior roots. We have hip flexion and ankle flexion. Then the next phase is weight acceptance. Weight acceptance is the quadriceps muscle. So then we are going to target the L3 roots, weight acceptance, and then finally you have to propel your body forward. So this is your calf muscle, the triceps that is becoming activated. That's another electrode at the bottom of the electrode array for the propulsion. And when you do this, you have reorchestrated the symphony of muscle activation during walking. This is how it looks like. This is David at the beginning of his training with the implant. Arc on means the electrical arc is turned on. you can see now he can walk with assistance. When it's off, can't walk anymore, but he immediately walk again when we turn the stimulation back on, exactly as you have seen in the rodents. And you have this complex stimulation pattern that you see on the left, on the electrode array. That's our biomimetic stimulation. And it was very important because, see, if we apply tonic continuous stimulation at the bottom, he's not capable of walking. So all the years we invested in understanding the mechanism of action of the electrical stimulation of the spinal cord was critical to reach this moment when David could take his first step after 8 years of paralysis. And when I say a first step, it's really like a voluntary control of walking. And I really want to emphasize this in this video, where we stimulate, we don't change at all the stimulation, but we ask David with his left leg, which is completely paralyzed, cannot activate a single muscle voluntarily, we ask him to make normal step and then suddenly to exaggerate the movement of the leg. But we don't change the stimulation. That's no is residual pathway from the brain that, because of the stimulation that amplify this residual pathway, allow him to exaggerate the activation of his hip flexor muscle in order to increase the step height. So we don't induce walking, we enable him to control the activity of his muscle with the residual pathway from the brain. And this is not only David. I put a lot of emphasis with David because I have this very long relationship with him, but as of today, Jocelyn has implanted 9 participants, 6 with what we say clinically incomplete spinal cord injury, so similar to David. They are well-share bound, but there are some residual pathway, but also 3 individuals who have a clinically complete spinal cord injury, meaning there is clinically no sign of any connectivity between the brain and the spinal cord that control normally walking. And actually, one of these 3 participants with this very severe lesion is with me today to show you a little demonstration and share with you his experience going through this clinical trial. So one of the 3 participants with a clinically complete spinal cord injury is actually with me today. So Michel, why don't you join me, turn on your stimulation, and come tell us about your experience in this clinical trial. Up, turn on the stimulation. Perfect. Welcome, man. So tell us, what have been your experience in this clinical trial? What does it bring to you, this stimulation?
Was emotional. I start again to walk, and was like a second opportunity.
Fa piacere. And so what's next for you? You spent a couple of months with us. So
yes, now it's only the beginning, and then I want to improve for using the stimulation in the normal life.
Of course.
Walking.
And you can imagine, like, Michel was completely paralyzed. He can stand with the stimulation, and stepping still can be improved a bit, but it's already a tremendous improvement, right? You can like stand for a long time like this. Last time we had a beer together, right? When you were standing. Yeah.
I was good because every day there is a goal that we have to reach, and we reach this goal, and we know that every small goals make a big difference at the end.
Yeah, and bring you closer to your goal to really have more independence, right, in your daily life. Cool. Well, thank you a lot. I'm really happy that you had a great experience with us, and we'll continue to follow you for many years. Don't worry, we don't give up on you.
Thank you.
Thank you so much for coming.
Au revoir.
So you have seen Michel, who was completely paralyzed for 2 years, And now when the stimulation is turned on, can take a few steps, can stand for a very long time. And the reason why it's so important is that now we can perform rehabilitation very active, meaning not like automatically in an exoskeleton when you have very few volitional activation of the muscle, but really intensively. And when we do this, what we observed was very surprising. But I will let David share with you this surprise that happened to him after 2 months of rehabilitation.
Ready? So Gonzalo asked me how long I slept last night. I said 6 hours, but it was 5. Because I woke up and checked the toes on the left.
Pour la première fois, il est capable de bouger légèrement sa jambe paralysée, sans aucune assistance, sans aucune stimulation.
And we just trained for 2.5 hours. And it still works. It even worked more before. Yep. Maybe some plasticity, huh? Let's see. Yeah.
So you can imagine it was a really emotional moment for us. David, for 9 years, was completely paralyzed on this left side, and suddenly he can move a little bit his toe. Doesn't look much, but for him it was a big first step. And after the additional month of training, it get better and better. Then he could do the little activation of his quadriceps, then a full leg extension against the direction of gravity. And then, after a few additional months of practice, he was able to take a few steps over ground without assistance, without any need of the stimulation. It was very surprising for us. Like, it's almost we did not understand how, after so many years of paralysis, you can observe such neurological recovery, suggesting that new nerve fibers were regrowing in response to this stimulation and training. So it was so new, so groundbreaking to us that we decided to publish these results. So they appeared in Nature, and the BBC decided to cover this event.
Good evening and welcome to the BBC News at 6. A man who was told he would spend the rest of his life in a wheelchair has walked again thanks to extraordinary work by scientists in Switzerland. The 30-year-old, who was left paralysed after a martial arts accident, has had an implant attached to his spine which boosts the signals from his brain to his legs. And incredibly, some of the damaged nerves have regrown. Another man who had a cycling accident has regained some mobility. Palab Ghosh reports from Switzerland.
So there was so much coverage of this result throughout the world that our institution here in Lausanne decided to support us, Jocelyn Bloch, the neurosurgeon, and myself, and launched a new center dedicated to this type of research. So it was, my school, the Swiss Federal Institute of Technology, the University Hospital of Lausanne, and we had a very important person here in Switzerland, Daniel Borel, who is the founder of Logitech, who decided to support this center with his foundation, the DefiTech Foundation. So it was amazing. This way, we were able to create what we call Neurorestore. Neurorestore, this center dedicated to have really in-depth understanding of the science and translate this discovery and technology all the way to application in daily life. And this philosophy is embedded in the little circle you have seen throughout my presentation. This is how we have different divisions that covered all the mechanistic work in mice, the amplification in rats, all the way to startup that commercialize this discovery. And you see that there is a bidirectional movement of the science. It's because we don't go only from mice all the way to humans, but for us going from unexpected discovery in humans back to the mice for the understanding, to then amplify this discovery is very important. This is our core philosophy at Neurorestore. And I would like to illustrate this philosophy and its implication with one example. I showed you that David recovered some movement without stimulation, despite years of paralysis, which was very surprising. So we modeled this recovery in rats. So at the top, you see a rat who have not been trained, he has this severe contusion, he's completely paralyzed. The rat at the bottom has been trained with a stimulation, and now he's able to perform these few steps, and it's not perfect, but there's a clear recovery of some stepping capabilities without the stimulation, suggesting that something has changed in his central nervous system to enable this recovery. And we wanted to understand why. So for this, we wanted to have this completely unbiased. So we developed the capability to render the brain and the spinal cord completely transparent. You can see through. We developed next generation microscope that basically combines sheets of lights that are passed through these transparent samples, so you can now visualize all the connection from the brain to the spinal cord completely unbiasedly, because we don't cut at all the central nervous system. We see the entire brain and spinal cord in one scan. You can see on this screen here how we label the projection from the motor cortex in blue, and in white you have the connection from our brainstem, the critical pathway to initiate walking. And with this, what we found is that this highly active training with willpower, with this enabled state of the spinal cord with the stimulation, trigger the growth of new nerve connection. So you can see all the nerve fibers here hitting the contusion, some retract, but some, you see, as spares, they escape the contusion, and where we stimulate, woof, you have this massive growth, like 1 to 5-fold increase in the density of projection. And it's really what David called neuroplasticity that enable the recovery. So that's one mechanism that we discovered with the animal models, the growth of descending pathways. But what we also found in our participants is that when they walk and we monitor the activity in the spinal cord, this is a PET scan, you can see that after the therapy, there's less activity in the spinal cord, which, I mean, also was very surprising to us, because you would expect that thanks to the training, they would activate more and more the spinal cord because it's become more efficient in performing the task. So it was so unexpected that we decided to go back to mice in order to really dissect what was happening in the spinal cord as the animal regained the ability to walk. And for this, we performed the first single nucleus RNA sequencing of the spinal cord of trained and non-trained mice. And here what you visualize are more than 80,000 cells ordered based on the expression of their gene. And we develop an artificial intelligence algorithm that is able to prioritize which of these cells are most perturbed, more responsive, meaning responsible for the recovery of walking after paralysis. And as you can see, it's really a little subset of neurons, very unexpected. So we tested their contribution. We have a transgenic mouse model that allows us to visualize. You see these neurons. You see this very small population of neurons in the middle of the spinal cord connected down the ventral aspect where you have all the cells controlling the muscles. And if you have a mouse that has not been trained, stimulation is not very effective, she cannot really walk. What we do in this animal is that we can just activate these neurons, just this small population of neurons. And when we do this, the stimulation effects are immediately augmented, and the mouse is able to regain this continuous locomotion, just targeting this one group of cells. It was really unexpected for us. And I think illustrates very nicely this importance of having all these different levels of understanding and ability to address very specific aspects of the mechanism, and then build technology that leverage this understanding to go all the way to humans. And that's the other direction. For us, it's very important that our scientific discovery, even in clinical studies, translate into medical devices that can be used throughout the planet by our participants to improve their quality of life. So for this, we developed first step of the solutions that involve a connected watch. So, there's a voice controller, you can use it with your hands that enable to tune the stimulation, which, embed many different sequences of stimulation. So you can do standing, you can walk, you can have cycling, you can optimize your trunk control because people with spinal cord injury have problems in the trunk. And all these sequence of stimulation that are controlled either by clicker, so you have seen before that actually Michel was using the clicker in order to facilitate this movement, or you can have motion measurement units that control this automatically. So patients, depending on their severity or their preference, they use different technology, but that's basically enable people like David to use the stimulation outdoor. We free them from the laboratory environment. And this is not only David. This technology are used by many participants to do many different tasks. So you see here Michel doing boxing while standing, David participating to the Wings for Life run. We have Gordon, who had very poor trunk control and loved canoe kayak, and now with the steam on, his trunk is stable and he can do paddling on the lake here in Lausanne. So, these are our first step. These people are not normal, they still are very injured, and you can see for Michel it's difficult to take a step. But it's one step, and each step is so important in getting toward the final goal to really regain some autonomy in the community for these people with disability. And for me, this journey from the paralyzed rat to a technology that is can begin to be used in people with paralysis to walk in the community was best embodied through a very emotional moment of my career. When I relived the dream that I formulated when I was a young postdoc. Remember Christopher is walking on stage. And this moment was during the charity in Geneva. We had 500 people who were donating for spinal cord injury research. And at the end of our presentation with Jocelyn, David surprised them and walked on stage. There we go. So you can see how easy it is for David to use the stimulation, turn it on, off. He can walk around. It's not perfect, but I think it's a very important step. And I want to believe that this Harvey Prize is celebrating this moment. For me, this is what really captures all this 20 years journey from the paralyzed rat to the first steps of David. And the reason why it really touches me to receive this prize is not only because it's a celebration of all the technology we have developed, and I'm really thankful to the IIT to give me this award, but I'm even more exciting that the prize is meant to be used to further innovate, to continue the journey and have an additional breakthrough. Because what I have experienced with David walking on stage, I want to leave it with people who cannot use their hands and arm anymore because of the lesion being higher at the level of the cervical spinal cord. This is even more dramatic than losing the ability to use your leg. And with this prize, our team at Neurorestore will try to restore some ability to grasp, move your hand through what we call a brain-spine interface. So the idea is to have an electrode array implanted at the level of the cervical spinal cord. It will be exactly the same kind of technology and concept, targeting the dorsal roots in order to modulate different regions of the spinal cord. And of course, controlling your hand, your arm, is infinitely more complex than moving the leg to walk, which is more automated. So your volition really needs to be embedded in the way we stimulate the spinal cord. Actually, we can do this. We have shown in non-human primate already that we can decode the intention from recording of the brain activity. And we have linked it to the stimulation of the spinal cord in order to build a digital bridge, a brain-spine interface that will restore volitional control of the stimulation. And for this, we are going to use this completely new technology. In a way, it's similar to what Elon Musk and Neuralink are designing, except that this technology has already been used in humans. This is a system that is implanted in the skull against the brain that enables to have wireless recording of brain activity that we will link to the stimulation. And just to tell you that this is not a dream, that we are really concrete and grateful to have this financial support because we are ready to go towards human application. And right next to me, behind this wall, there is our upper limb lab, the same that we have used for walking, but not dedicated for the upper limb, where the clinical trial will take place early next year. So we are really excited, and all the team is so grateful for this prize and the possibility to launch this new line of research that is so important for people with spinal cord injury. So we'd like to thank you, and I would like to leave you with a 1-minute movie that just captures all the people, the emotion that we experienced through this 10 years journey.
Each individual going the way that we did here and investing the time and the energy we did is really hard. I mean, hope that others can profit from it. Let's hope and go for it.
Oh my goodness me. Well, welcome to the Q&A. Welcome, G. What a fabulous presentation. I'm not the only one who had tears in my eyes with that, I'm sure. Absolutely phenomenal work. Absolutely phenomenal. Congratulations. Okay, so we have lots of questions, so I'll start very quickly. First one, would this technique work on people that have been injured a long time ago, so several years ago?
Yeah, it's an excellent question. Actually, the real question is, will it work better in people who have been injured recently? Because all the data I've shown so far in humans are from people who have been injured for many, many years. So these are all what's called a chronic injury, when there is normally less possibility for recovery. So it was surprising for us to see so much what we call neurological recovery, meaning recovery without the stimulation. We do know in rats that if we start at 1 week, we have a fantastic recovery. If we wait 2 months, the recovery is much less. So if anything, it should work much better when we start very early. And this is a clinical trial that we'll do next year. We are preparing this in a large number of people to demonstrate that we can really have a true impact on the recovery with recent injury.
Amazing. That's great. Okay, thanks, Jay. Second one, we have at least one viewer who's worried about the lab rats. How are the lab rats now?
Yeah, it is always a very sensitive research. Of course, we are using a lot of animals. So we are doing our best to treat the animal in the best possible way. Also because it's very important scientifically. You know, they undergo rehabilitation procedures for a long time. So we spend many, many weeks, months with them, training them daily. So there's really this strong interaction with the physical therapist for rats, I would say. You know, so if they are not treated well, they don't engage in the rehabilitation, they don't try to recover. So we try to treat them as humanly as possible.
So it's humane and it's in your interest from a scientific point of view. Correct. As well. Yeah, great. Okay, thank you. Third question, are there any batteries for the electrodes to work?
Yeah, I went a bit faster on this part of the actual technology, right? I'm sorry. So you have the electrode array, which I emphasized a lot during the presentation. It is connected to what's called an implantable pulse generator. It's kind of a pacemaker that I use, you know, for heart therapy. So it's like a big, like a small can, basically, that is in the abdomen of the patient, and is, you know, battery powered, so it lasts for many years. And the beauty of this system we are using is that it's communicated wirelessly and in real time with an external computer. That will enable us to do this real-time stimulation. So it's a beautiful piece of engineering.
It is a beautiful piece of engineering. Wow, incredible. Great, thank you, G. Fourth question, can the same technology be used to regain arm and hand functions on a tetraplegic?
So this was like, I imagine at the beginning of my presentation when someone is asking, oh, can we also apply the same for the upper limb? And as you notice at the end of my talk, I really emphasize how with the Harvey Prize, we are going to jump into this new avenue of research and testing the idea of stimulation for the recovery of the upper limbs. And we really have a lot of data in preclinical models. So we are confident that we will have an effect, the extent of which is unknown, but we feel very confident that there will be a therapy to improve a bit the upper limb recovery at least.
Brilliant. That's good to hear. Thanks, G. Okay, fifth question. Is there an application for less serious or partial damage in the spinal cord? So, for example, where muscle wastage may be happening instead of paralysis?
So, of course, it's a very interesting question because it relates to the severity of the injury. How many nerve fibers are spared by the injury? And then what is the effect of the treatment? And clearly, there is a correlation between the amount of spare tissue and how well people recover. So the less severe, the better the recovery. The principle remains the same. So to respond to the question is that, yes, there is an application because in any case, when we stimulate, we improve the residual capacity. So if there is more, like paresis and not paralysis, then there is a better recovery. The more severe the people, the more they are dependent on the stimulation to have any sort of recovery.
Brilliant. Okay, great. Does the system need to detect signals from the brain or only simulate the nerves to respond to the residual signals?
Yeah, interesting question. So that's what I kind of touched also at the end. So for the leg, we have different ways to control the stimulation. So we stimulate the spinal cord, we activate the nerve fibers. The stimulation can either be controlled via a captor, wearable sensor that we attach to the feet, and that's sufficient to detect what the people are trying to do. Standing, walking, stand, swing. That's sufficient for basic function, like walking. Of course, if you have a more difficult execution, even for the leg, but even more, of course, for the upper limb manipulation, then it's probably insufficient. This is why now we have developed this brain-spine interface. It detects the intention from the neuronal activity of the brain and link it to the stimulation. And this, we did it in preclinical model, it works very well. And we are going to do this both for the leg and for the upper limb in the coming clinical trial. That would be the real breakthrough. That's what Elon Musk wants to do, basically.
Right, really? Okay.
Correct. And we have all the technology ready and validated by the ethical committees, so we are ready to go.
Wow. It does. It just feel like stuff of the future, doesn't it? Which is, the future is here. It's incredible.
Yeah, that's science fiction. Of course, we always need to calibrate, the expectation, because as you see, our patient, they take steps, but they are not cured. They really depend on this technology. It's very difficult for them still. It's a lot of effort, but that's one step toward the direction of a treatment that at least can improve the quality of life of these people.
Yeah, incredible stuff. Brilliant. Thanks, Jay. Okay, next question. Is the relative timing of the simulation controlled by internal or external electronics? And the question is, given that the simulation is switched externally.
Yeah, of course, this is related to the previous question in a way. And there are many ways to do it. As I said, we can even do what we call open loop. It means you have a pre-programmed sequence of stimulation, and the participant are able to time their desire to move to the stimulation. They know when the extension, flexion, etc. That's one way to do it. Second way is with this wearable sensor. So in this case, you have external electronic to detect the ongoing movement that then communicate wirelessly with the implanted pulse generator, the pacemaker in the abdomen. And the last step is brain signals.
Okay.
So we're exploring all possibilities, and depending on the severity and the type of patients, even their preference, they use different type of technology.
Oh, so the patient can have a preference?
Yes.
Wow.
Yes,
okay.
absolutely. Yeah. Some they like open loop, some they like the wearable sensor, some they like clickers. You have seen Michel, when he came to me, he was pressing on clickers that triggers a different sequence of stimulation.
Yeah.
And it depends. Some patients like it, some other, they all have their preferences, so we give them options.
Brilliant. That's so nice that you give them options as well. I guess you've answered the next question then. So can a similar rehabilitation effect be gained from patients using wearable electrodes?
No, it's actually a different question, and very interesting as well. So there is the idea that you can use transcutaneous stimulation to stimulate the spinal cord. In this case, you do not need a surgical intervention, which is indeed very interesting. Some people can be afraid of surgery, very rarely, but you can have complication, right, with surgical intervention. So we have been also exploring transcutaneous stimulation, and others have been doing this also in the United States. And now with the startup that we funded, Onward, there is a pivotal clinical trial in the US to test transcutaneous stimulation to improve recovery of the spinal cord injury. So I hope it will be successful, and this will be one option. Of course, less effective than an implanted solution, but still maybe sufficient to improve the recovery. That's what we are testing right now.
You're testing out now?
Right now. It's
awesome.
ongoing in the US.
Amazing. Good stuff. Okay, next question. Who qualifies for the study? Are there any limits, such as the American Spinal Injury Association impairment scale, C4 and C5, where the stimulation cannot work?
So there is only one limitation in terms of the location of the injury, is when the injury is located where we want to stimulate. For example, to regain walking or mobility, you have to stimulate we have to stimulate the 5 centimeters of spinal cord in the lumbar region. If the lesion is really in this region, then the neuron that we want to stimulate are dead. Then it's not effective. If the lesion is above, even at C4, C5, C2, the stimulation will still be effective to modulate the lumbar region of the spinal cord. Now, there are other problems though. If you have a very severe lesion, very high cervical, then you have no control of the trunk. There are many additional aspects that are affected and that will limit the ability to use a stimulation. But then for this type of injury, we focus more on the upper limb, on the trunk control, which is also a clinical trial that we have started right now to control the trunk, and blood pressure, because there's a lot of problem with hypotension when you have this type of high cervical injury. And we just published a few months ago a paper in Nature with a new stimulation approach to fix the problem of orthostatic hypotension together with the trunk control. Actually, we are recruiting participants right now. So if you know individuals who have hypotension issues after spinal cord injury, please contact me because there is enrollment.
Oh, amazing. Yeah. I mean, this is just sort of engineering, science fiction fusing together here, isn't it? You know, it's
yeah, we are, you know, spinal cord injury, I decided to dedicate my life to developing treatment for these people, and now we have sufficient understanding and like this technology that are available to really try to target many neurological functions. So we have many clinical trials now that we are launching with the hope that it will translate into therapy available throughout the world for people with spinal cord injury.
Yeah, just amazing work, Jay. Absolutely inspiring.
Thank you, Dan.
We've got a few more questions if you have time. Do the electrodes have a long life?
Yeah, so this type of electrode, the one we use in human, it's medical grade technology. So we must provide proof to the regulatory authorities that they will last for at least 10 years in the human body.
10 years.
For commercialization, absolutely.
10 years. That sounds like a long time. Is it a long time for you?
No, this is with existing technology. It's an expected lifetime. So there are many, you know, type of electrode, similarly for pain treatment, and they are reasonably resistant. Those happen that they break sometime, but in general, 10 years is reasonable. It would not be with a very innovative electrode that we used in rats, you know, the one that stretch, etc. Maybe that was the question. This one, of course, are more fragile.
Okay, but the ones in humans are 10-year lives.
Correct.
Yeah, cool. Thanks, G. Do you think in the far future, spinal cord injuries will just heal like a broken bone?
Well, I cannot predict the future, but I'm working with all my team at Neurorestore to make this a reality. You know, and it's still my career objective to have someone working in the hospital, paralyzed, and walking out of it. And I will not stop until we have achieved this objective.
Brilliant. What drives you, G? What's the inspiration and the motivation for you and the project?
You know, for me, it's all starting with a love for movement. Sport was central in my life when I was young, teenager. I was in high-level sports. That's why I decided to study how the brain controls movements. And through this research experience, I was confronted with people who lost the ability to move, which for me is so important. It was unbearable. At the time, I went to Los Angeles. I was working with young people of my age, paralyzed. Christopher Reeve was still alive, so it was a true, strong inspiration to guide my future step in research. And ever since, it only got stronger.
I'm glad it did. I'm glad it did. G, do you get a chance to talk to sort of the next generation of scientists and engineers about your work and inspire them to make sure that we have that pipeline of people still working on this?
I think this is a fantastic lux, I don't say in English, the opportunity, the chance I have in life that Neurorestore, you know, is 70 individuals, engineers of various expertise, neurobiologists. I mean, really, kids are so smart. You know, and the greatest thing that can happen to you is to work with people who are smarter than you. And this is the case. I mean, in this environment, they are amazing, these kids. and you can motivate them, they're full of energy. And this is why I'm still hopeful that we will achieve this breakthrough in the treatments, because we keep innovate, and you have such multidisciplinary combination of expertise that it might be possible that one day we do not only all this engineering that I'm showing you today, but also we're working very strongly on biological repair. We're growing nerve and combining them together so that one day we will have this type of therapy in humans. And this is what I have to do every day, inspire these people to work harder to achieve this feast of engineering and biological therapy development.
Brilliant. Well, I'm glad you do spend some time doing that as well. And I'm sure it's from my point of view, you are entirely inspiring. So I'm sure lots of that next generation will find you very, very inspiring to talk to.
Thank you, Dan.
Thank you so much. That concludes the questions that we have as well. You are such a difference maker, G. Thank you so much for joining us today as well and making time, and many congratulations on your
I appreciate it. So thank you so much for the prize, and I will make sure that we are conducting a research that is worth of this reward. And thank you so much.
And we'll keep an eye on what's going on and make sure we update all of our members and all engineers across the world on.
Feel free to invite me to London. We are enough of these video interactions, right?
Definitely.
Digital world need to stop.
Yes, a nice show in London. Good presentation in London. We'll put it on the list.
Wonderful. I would love it.
Thank you so much,
Thanks a lot, everyone.
Thank you very much, everyone, for watching. I'm sure you are inspired as I am from G's amazing work and his presentation. Just a reminder that we have the IET 150 show, which will be on our YouTube channel at 1 o'clock as well. So a festival of engineering for you today. Thank you very much, everyone, for watching. Bye-bye.
Bye-bye. Thank you.
Thank you so much.